Hello and welcome to another Macular and Me webinar, which has a working age focus. As you know, we now record these and post them on YouTube and the Macular Society YouTube channel and website on the last Thursday of each month. So there's a huge back catalogue to have a look at as well, which you can find in the same place. We've also started creating podcasts off the back of these, so you can listen to them at your leisure too. So that's all very exciting stuff. We've got a returner this time, which is great. So we funded a bit of funding over a couple of years ago for a researcher, and she's coming back to talk about her results of the research that she's undertaken. We'll introduce her in a second, but just to say, I'm joined as usual by my colleague Sarah Clinton from the Macular Society. So how are you, Sarah? You okay?
I'm good, how are you?
Oh, you're not too bad, not too bad. Now, so Sarah, I'm sure will be asking some questions at the end of this of the session as well, and I'm sure I'll chip in a couple as well. So there we go. So no, without further ado, let's introduce our presenter for today. So Jackie van der Spuy. So how are you?
I'm fine, thank you, Colin. And you today?
Oh yeah, not too bad at all. Not too bad. I'm looking forward to hearing about all your research and what you've been up to over the last couple of years since the last time you joined us. So I'm sure you've been incredibly busy. So yes, so it'll be interesting to hear what you've been up to. So great. So I'll leave to introduce yourself and for you to share your screen and off you go.
Thank you so much, Colin. So I'm Jacqueline van der Spuy. I am a professor of molecular and cellular biology at the UCL Institute of Ophthalmology. And my research interests are on in disease mechanisms of inherited retinal degenerations, particularly inherited retinal degenerations affecting younger people from juvenile to early adulthood. So I think without further ado, I'm not sure I have a lot of time, so I'm going to kick off and share my screen with you and go straight into my presentation. And so today I'm really delighted to be speaking to you about a project which has involved the development of an antisense oligonucleotide therapeutic for Doyne honeycomb retinal dystrophy, which has very kindly been funded by the Macular Society for the last couple of years. And so it is really exciting to give you an update on this particular project. So Doyne honeycomb retinal dystrophy is a juvenile macular dystrophy, so it affects young people, and visual impairment or the onset of visual impairment is variable, but it's generally around about from the fourth decade of life. And the disease follows a autosomal dominant Mendelian mode of inheritance. And so this means that in order for an individual to manifest the disease, they need inherit only a single copy of a faulty gene from either the mother or the father. And this means that the offspring have a 50-50 chance of inheriting the faulty copy of a gene and therefore manifesting the disease. And this is compared to autosomal recessive disease, where in order for an individual to manifest the disease, they need to inherit a faulty copy from both the mother and the father, and so they have only a 1 in 4 chance of manifesting the disease. If a child inherits a faulty copy from either the mother or the father, then they are a carrier and they don't manifest the disease. So yeah, Doyne honeycomb retinal dystrophy is an autosomal dominant disease, so it's passed from parent to child, and it affects an area of the retina known as the macula. So I'll just give you a little bit of an overview of the retina itself. The retina is the neurosensory part of the eye. It's the bit that senses the light. And over here you can see, I wonder if I can get a pointer. Over here, you can see a histological section of the retina, and you can see that it is an exquisitely organized bit of the central nervous system and the most accessible bit of the central nervous system. And it consists of 3 layers of cells: the outer nuclear layer, an inner nuclear layer, and a ganglion cell layer. And the outer nuclear layer consists of a group of cells called photoreceptor cells. And as the name implies, these are the photosensitive cells of the eye. They're responsible for converting a signal of light to an electrical impulse that is passed onto the brain. And in this schematic over here, you can see that there are 2 types of photoreceptors in the human retina: rod photoreceptors and cone photoreceptors. So rod photoreceptors are responsible for our dim light vision, so we use them at dawn and dusk, whereas the cone photoreceptors are responsible for our color daylight vision. And both the rod and cone photoreceptors have a very special compartment called an outer segment. And the outer segment houses the machinery which is required for that conversion of a photon of light into an electrical signal. And the rod and cone photoreceptors sit on top a very important layer of cells called the retinal pigment epithelium. Now, the retinal pigment epithelium is a monolayer, a single layer of cells, which forms a very tightly packed barrier between the retina and between the choroid. So the RPE, the retinal pigment epithelium, or RPE as it's called, forms the blood retinal barrier, if you like, which is akin to the blood-brain barrier. And it plays an incredibly important function. So it's responsible for transferring metabolites to and from the retina to the circulation. It is also plays a critical role in the visual cycle. So it's involved in the recycling of 11-cis retinal, which is the chromophore, which is attached to the light-sensing pigment in the photoreceptors. And you can see over here on the apical side of the RPE, there are a number of microvilli which extend between and around the outer segments of the photoreceptor cells. And the photoreceptor cells undergo a diurnal shedding of these outer segments and a daily renewal of outer segments from the base of the outer segments. And so the RPE cells play a very important role in engulfing those photoreceptor outer segments. So they really play a very critical function. And in Doyne honeycomb retinal dystrophy, as I mentioned before, it's a juvenile macular dystrophy. And so the area of the retina that's affected is an area called the macula, part of which is the fovea. Now, this graph over here shows a cross-section through the macula, including the foveal area. And essentially, the graph shows this distribution of cone and rod photoreceptors from the center of the retina, which is towards the back of the eye, and as you progress towards the periphery of the retina, which is as you move forward from the back of the eye towards the lens and the pupil. And what you can immediately see from this graph is that in the fovea, the cones are distributed at their highest density at the exclusion of any rods whatsoever. And then as you move from the center to the periphery, the cones rapidly drop off in number. And just around the edge of the fovea, the rods peak in number, and the rods too then drop off in density as you move from the central to the peripheral retina. And where the optic nerve exits the back of the eye is where you have a blind spot. But essentially, the fovea is the bit, or the macula, including the fovea, is the bit of the retina which is responsible for our daylight color vision, because this is the area where these cones are packed at their greatest density at the exclusion of the rods. And so this juvenile macular dystrophy therefore manifests clinically as a syndrome in which the central vision mediated by cones is primarily affected. And in patients, this can manifest as blurry central vision, colors may look dull, patients might see blind spots, and straight lines may also appear curvy. So what is the genetic cause of Doyne honeycomb retinal dystrophy? It's a mutation in a gene called EFEMP1. And the gene EFEMP1 in turn codes for a protein called fibulin 3. And fibulin 3 is a member of a group of proteins called fibulins. Now, fibulin 3, like all the members of the fibulin family, is actually a protein which is produced inside cells, and then it is released from cells to the extracellular matrix. And it's thought to play a very important role in the remodeling of the extracellular matrix. And so the actual Doyne honeycomb retinal dystrophy, another clinical feature is the formation of drusen. So what are drusen? So drusen are clumps or accumulations of lipids and proteins which form underneath the RPE layer. So between the RPE layer and the choroidal circulation, there is a membrane called Bruch's membrane. And in Doyne honeycomb retinal dystrophy, we have the accumulation of lipids and proteins underneath the RPE, between the RPE and between Bruch's membrane. And in the image over here, you can see a fundus photograph of these drusen, which you can see here first begin in the macular area, and then they spread out radially in a honeycomb sort of pattern, hence the name of the disease is Doyne honeycomb retinal dystrophy. So we have these drusen accumulating primarily within the macula and therefore affecting the central vision. We understand that the RPE in the macular region become compromised and therefore affect the function of the overlying photoreceptors within the macula. Now this just shows another image of a patient with Doyne honeycomb retinal dystrophy. This image over here is known as has been taken using a imaging technology known as optical coherence tomography. It's a non-invasive imaging procedure where light is shone into the eye, and you can see the hyper-reflective layers of the retina. And over here you can see the fovea, and the top image over here shows an OCT image from an unaffected individual, and the panel beneath shows an OCT image from a patient with Doyne honeycomb retinal dystrophy. And I hope you can appreciate over here in the patient, you can see these pillowy clumps or drusen, which are not present in the unaffected individual. So the aim of our project was to develop a novel therapy to treat Doyne honeycomb retinal dystrophy, because obviously at the moment the condition is incurable. Some patients do develop a choroidal neovascularization, and the treatment for that is anti-VEGF therapy or laser therapy. However, it doesn't halt the progression of the disease. So there is actually no treatment or cure for the disease at the moment. And in this project, we have used two state-of-the-art kind of technologies, namely stem cell technologies and gene editing technologies, to achieve the first goal of this project, which was to model the disease in vitro. And so how do we do that? So to model the disease in vitro, we take somatic cells from patients. In our case, we use renal epithelial cells, and these are cells which are naturally shed off in the urine. You can also use cells from the blood, hair cells, or cells from the skin, but we use renal epithelial cells. And then we reprogram these renal epithelial cells into so-called induced pluripotent stem cells using a number of very special factors called Yamanaka factors. And induced pluripotent stem cells are a very powerful tool because, as the name implies, they can form any other cell type in the human body, or they can be induced to form any cell type in the adult human body, given the right growth conditions and factors. So in our project, we've taken renal epithelial cells from two unrelated patients with Doyne honeycomb retinal dystrophy, reprogrammed them to induced pluripotent stem cells, and then used the right culture media conditions and small molecules to differentiate them into retinal pigment epithelial cells, which are the cell types affected in this disease. And this is a very powerful technology because the retinal pigment epithelial cells we are able to grow are genetically identical to the patient from which they have been derived, including the fact that they harbor the disease-causing variation and are therefore expected to manifest the disease or disease characteristics. So we then have used a second powerful technology called gene editing technology to engineer our induced pluripotent stem cells that we have obtained from the patient. So I'm sure many of you have heard of CRISPR-Cas9 gene editing or these molecular scissors. The idea of CRISPR-Cas9 gene editing is that you can use these molecular scissors to direct an enzyme to a very specific location in the genome and create either a single or double-stranded break. Now, when this happens, there's two pathways by which this double-stranded break in the DNA is repaired. One is called non-homologous end joining, and the other is called homology-directed repair. So in non-homologous end joining, what happens is that a little patch of nucleotides or DNA bases are either added or removed to try to repair the double-stranded break. And this is actually a very deleterious process, and it actually leads to gene disruption. In our case, we used a technology called homology-directed repair, which uses a donor template, which can precisely repair a genetic variation or introduce a genetic variation at a target site of interest. And so we use this technology to repair the genetic variation in the patient iPSC lines. So over here in the top panel, this is a sequencing chromatogram, and the arrowhead shows the identity of the nucleotide base at this position. And you can see there's 2 peaks, and this indicates that the patient is heterozygous. They have a copy of the wild type gene, and they have a copy of the gene harboring the variation, which can be seen here by these 2 peaks. And we use this gene editing technology to repair this variation. And so in the bottom chromatogram, you can see the wild type sequence only. So we use this technology to generate what we call an isogenic control. And isogenic controls are very important in disease modeling experiments because they control for any other kinds of variation, for example, due to genetic, epigenetic differences, etc. So it's a really good control when you are performing disease modeling experiments. So after we have our iPSC lines, the next step is to differentiate them into retinal pigment epithelial cells, and we use an established protocol to do that. So this is just a couple of images of the retinal pigment epithelial cells that we're able to produce. And this image over here in the top left is looking at the RPE cells from above. And you can see that they form a very tightly packed single layer of cells, which are packed together, and they are pigmented and they have a cobblestone appearance. If we look at these cells in cross-section, so we cut through them, you can also see that they're polarized. So they have certain things which are expressed at the basal surface and other markers which are expressed at the apical surface, and they are pigmented. So they're packed full of melanosomes, which give them a pigmented appearance. So this over here is an image to show RPE cells that we have differentiated from our repaired control at the top and from our patient at the bottom. And we can use a technology called immunofluorescence laser scanning confocal microscopy to look at certain features of our cell types in culture. And over here at the top, you can see again using this green marker that our cells, both in the patient line and in the corrected line, form this monolayer of tightly packed hexagonal shaped cells, and that both lines express PMEL, which is a melanosome associated protein. And this was 60 days after differentiation. But if we leave our cells even longer, the patient cells begin to manifest characteristics of the disease in comparison to the repaired control. So for example, over here we can see disorganization of the melanosome associated protein. And another important feature we've looked at in our disease model are proteins that are associated with the lipoproteinaceous accumulations that occur in disease. So in juvenile macular dystrophies like Doyne honeycomb retinal dystrophy, as well as in age-related macular dystrophy, for example, all of these diseases manifest the accumulation of drusen, and the drusen themselves accumulate, as I said, a number of lipids and proteins. And one of the proteins which is typically found in these drusen is collagen 4 or COL4, shown here. And what you can see is that as our control cells and our patient cells differentiate, the patient cells begin to manifest the accumulation of this collagen 4 underneath the RPE layer. Another important marker of drusen or subretinal deposits is another protein called apolipoprotein E. And again, apolipoprotein E, shown in magenta here, is a typical protein which accumulates or is associated with drusen formation in juvenile macular dystrophies and age-related macular degenerations. And again, in our patient line, we can see accumulation of this ApoE protein underneath our RPE layer compared to the control. And again, if we look at these, the patient line and the control line in cross-section, so we section through it and we look through it in cross-section on face, you can see, first of all, that for the control line, you get a really nicely organized monolayer of cells, which are highlighted here in blue. The blue labels the nuclei of these cells. And underneath these cells, you can see a nicely organized organization of both fibrillin or EFMP1 and collagen, which is shown in magenta. But in the patient cell lines, you can see that you get a large accumulation of collagen 4 underneath the cells, which are somewhat disorganized localized, and you also see a patchy accumulation of this fibrillin as well. So in our disease model, we have been able to show that we are able to manifest some of the disease characteristics that are found in patients in vivo. So this is therefore a powerful tool that we can use to test therapies. So the next aim of the project was to see whether we could develop a novel therapeutic for Doyne honeycomb retinal dystrophy. And the therapy we chose to focus on is an RNA-directed therapy, namely an antisense oligonucleotide therapeutic. And so to explain how that works, I just will explain a little bit about our central dogma of how DNA is used to code for the synthesis of a protein. So each of us in our nuclei, we have double-stranded DNA. And the first step towards making protein from the DNA is to transcribe a strand of RNA, which is ribonucleic acid. The RNA itself is then further processed to form messenger RNA. And in this process, the non-coding bits of the RNA are spliced out, and the coding bits of the RNA are spliced together to form something called messenger RNA. And the messenger RNA, in turn, is translated into the protein. And this is how your DNA codes for protein within your nuclei. And the therapy that we've developed targets the messenger RNA strand within the cell. So you can imagine that within a cell, you will have a messenger RNA which has arisen both from the wild type copy of the gene and a messenger RNA which has arisen from the mutant copy of the gene. And our strategy was to try to target the mutant copy of the gene. If we can target the mutant copy of the mRNA, then we can prevent the formation of the faulty protein. And an antisense oligonucleotide therapy achieves that by targeting the messenger RNA. So an antisense oligonucleotide is a small piece of DNA which is complementary to the strand of mRNA that you wish to target. And in our approach, we used a microwalk approach to design a number of antisense oligonucleotides. And as I said, these will bind to the messenger RNA, and this forms a DNA RNA heteroduplex, which is recognized by an enzyme called RNase H, and the RNase H will chop up the bit of messenger RNA. And as I said, in our approach, the idea was to design an antisense oligonucleotide that could specifically target the messenger RNA arising from the faulty copy of the gene, thereby chopping up the messenger RNA and preventing the formation of a faulty protein. So the way we did this was, first of all, to use a microwalk approach. We designed a large range of antisense oligonucleotides which spanned across the mutation site. And we first used what's known as a heterologous expression system. It's just a very fancy technology for saying that we engineer the messenger RNAs and we artificially introduce them into an irrelevant cell type, and then we treat with the antisense oligonucleotides and monitor the levels of the resultant transcript. And the slide is a little bit complicated, but just to show you over here at the bottom, this just illustrates 4 different antisense oligonucleotides that we tried. And in pink, you can see one antisense oligonucleotide, antisense oligonucleotide 1, that significantly reduced the levels of the transcript. In comparison, you can see here that the antisense oligonucleotide shown in green didn't work. And we're showing these levels compared to the bars shown in white and gray. So once we identified an antisense oligonucleotide that could work reasonably efficiently, we made further chemical adjustments to the antisense oligonucleotide. It's worth noting that antisense oligonucleotides are very unstable in cells. And so when you engineer an antisense oligonucleotide, you introduce a wide range of different kinds of chemical modifications which try to increase and improve the specificity, the bioavailability, and the efficiency of the antisense oligonucleotide. And so we made a number of modifications to this ASO1. Over here, they are depicted as ASO 1.1, 1.2, 1.3, and 1.4. And the take-home message of this slide is that you can see, for example, in blue, that we get a concentration-dependent decline in the transcript levels. And this appears to be primarily mediated by transcript arising from the faulty variant of the gene. However, just to reiterate that this is a heterologous overexpression system, so we have introduced the messenger RNAs into any irrelevant cell type. So once we were able to find an ASO that we thought was efficient, the next step was to ask whether it would work in our iPSC RPE disease model. So in this case, we used an assisted delivery system to deliver the antisense oligonucleotide to both our isogenic control and to our patient RPE line. And we used a technology called next generation sequencing, which can very accurately quantify the levels of transcript arising from the normal copy of the gene and from the wild type and from the mutant copy of the gene. And these genes are obviously endogenous to these cells. So we're not introducing anything into these cells other than the antisense oligonucleotide. So over here, this shows the levels of transcripts in the renal epithelial cells that were derived from the patient. And you can see that in a patient, they have shown in light blue and dark blue, they have about a 50% level of the transcript from the wild type gene and the transcript from the faulty gene, which is as expected because you inherit one copy from the mother and one copy from the father. And similarly, with the cDNA, you see a 50-50 distribution of the transcript arising from either the wild type copy of the gene or the mutant copy of the gene. If we look at that distribution in the iPSC RPE model, we see there is a little bit of allele skewing. In other words, we see a little bit more of transcript from the wild type copy of the gene. But if we treat these iPSC RPE with the antisense oligonucleotide, you can see there is a massive reduction in the transcript from the mutant copy of the gene, with almost the entire amount of the transcript being accounted for by the wild type copy of the gene. So what this data shows is that we've been able to effectively design and deliver an antisense oligonucleotide, which can very effectively target the mutant allele, leaving the wild type copy intact. And how does this then translate to the actual disease model itself? We then also went on to treat the disease model with the antisense oligonucleotide, but we did this gymnotically. So in the previous experiment I showed you, we used assisted delivery to deliver the antisense oligonucleotide to the cells. Over here, this is naked delivery of the ASO to the cells, which is referred to as gymnosis. And it's much more relevant to a clinical situation where naked ASO would be injected intravitreally into the eye for treatment. So if we just take naked ASO and we plop them onto the cells, you can see in this bottom panel over here by green fluorescent that there is a very efficient uptake of the antisense oligonucleotide by these cells. And this is just an antisense oligonucleotide, which is tagged with a little fluorescent tag so we can see how and where it's taken up. And just to mention that these RPE cells are professional phagocytosing cells. So I mentioned before that one of their roles is to engulf photoreceptor outer segments. So they're actually pretty good at taking up stuff. And so over here, we can see in the top panel that in the isogenic control, there's very little accumulation of ApoE. Reminder that ApoE is apolipoprotein E, which is a protein associated with drusen in these diseases. In the patient line, we can see significant accumulation of this ApoE, and in our ASO-treated line, we see a significant reduction of ApoE accumulation. So this seems to indicate that the antisense oligonucleotide can ameliorate some of the disease characteristics in a patient-derived disease model. And so just to go over the conclusions in brief, what we have done is we have developed a relevant patient-derived model of Doyne honeycomb retinal dystrophy that can manifest characteristics of the disease compared to an isogenic control. Second of all, we've shown that we have been able to develop an antisense oligonucleotide therapeutic, and that this therapeutic can specifically target the disease-associated variant, and that it can moreover reduce the disease phenotype in the patient-derived model. And so at the moment, what we've done is we've applied for a patent for this antisense oligonucleotide therapeutic, which is under consideration at the moment. And of course, the next step would be to see if we could advance this therapeutic towards the clinic. And there are various challenges and limitations with respect to that. But just to conclude, I would like to sincerely acknowledge the Macular Society. I'd like to thank them sincerely for supporting this project over the last couple of years, and it's very exciting to see where it will go next. I'd also like to acknowledge the postdoctoral research fellows who did a lot of this work, Dr. Olivia Rezek and Dr. Bea Sanchez-Pentado, as well as a number of people that I've collaborated with at the Institute of Ophthalmology. And with that, I'd like to say thank you so much, and I would like to ask if there are any questions.
Jackie, that's wow, what an amazing piece of work you've been undertaking over the last couple of years, and it's quite incredible. So thank you for joining us and letting us know how you've been getting on.
Thank you.
Sarah, have you got any questions? I'm sure you've got a couple.
Yes, no, that was a really interesting explanation about the project, how it's all gone. You did cover it right at the end, but so what would the next steps be in order for this to ultimately, hopefully, become a treatment for patients?
Yeah, so I mean, it is tricky. The translational pipeline is very difficult to navigate when you come from fundamental science background. And the first step of that is obviously to see if we can patent the technology to protect the IP associated with this. Fortunately, there is always a sense of commercialization associated with a therapy like this. But the next step would be to see, one of the things would be to obviously license the technology to a company who would then have sufficient funds to take it forward. If you weren't able to do that, the other thing would be to search for a much bigger pot of money to see if we could enter this into, the translational pipeline, which would involve, preclinical testing of various characteristics of the antisense oligonucleotide. So, for example, its toxicity, for example, its pharmacodynamic profile, when applied to an in vivo situation, those sorts of things. Just to say that, of course, in terms of advantages of where we're at, is that the ASO technology for ophthalmic conditions is actually quite well characterized. There are a number of antisense oligonucleotide therapies which have been delivered intravitreally for the treatment of retinal disorders. So there's one called sepofarsen for LCA10, there's one called ultevursen for Usher syndrome, and there's one in development for autosomal dominant RP. And the other thing is that the intravitreal delivery of ASO to the eye is well established as being safe and effective. So, that's one advantage, or that's one of the positive aspects of trying to take this along the translational pipeline. The other big challenge, though, is that Doyne honeycomb retinal dystrophy is a rare disease. And so therefore, with funding, you're probably looking possibly at philanthropic funding for a rare disease, etc., because, when these things enter into a translational pipeline, there's the challenge of having a large enough cohort to conduct the appropriate kind of safety tests. And that's hard to do when you're looking at a rare disease. So the next stage for us would be to look at further funding to progress this along a translational pipeline, to look at some of those, to look at the tolerability, toxicity, and the other properties of the ASO in an in vivo environment.
So yeah, it's really, really interesting, and good luck with all of the next steps.
Thank you.
So with this ASO treatment, is that so intravitreal injections into the eye? So what would that be looking like for patients? How often would they be having to have these treatments, or is that all a bit dependent?
Yes, precisely. It's a very good question. In fact, in the next step of our project, if we're able to get further funding from it as well, is to, so in our model, we have the opportunity to ask the question of whether the antisense oligonucleotide can prevent the formation of these subretinal deposits altogether, or if it can resolve the formation of these deposits after they have formed. And that's an important question because many patients will come to a clinic or attend a clinic only when their vision is affected. And by that stage, the drusen have already formed. So an important question is, if we treat patients once the drusen have already formed, will there be a benefit? Will there be a beneficial outcome for them? So can the ASO revert the formation of these drusen and enable a halt in the progression of the degeneration is an important question. As I said, many of the families have an autosomal dominant inheritance. So, many will already know from their parent or whatever that they may be a carrier, maybe at risk of developing the disease. So perhaps they might come to the clinic earlier before the onset of drusen, and that might be the time to treat. But much depends, on when patients come to the clinic. And at the moment, it would be an intravitreal delivery. As I said, that's very well established delivery route for antisense oligonucleotide technologies for ophthalmic disorders. However, it has become clear that, first of all, repeat treatments are required when you have intravitreal injections. Intravitreal injections are quite common for other kinds of treatments as well, like anti-VEGF treatments, etc. But it is quite invasive. And the other thing is that there is the risk of cataract formation with repeated injections. And one of the options that people are looking at going forwards is to try to explore other modes of delivery other than the intravitreal injection, like suprachoroidal, for example. So there is a long way to go, but there are many, many, many clinical trials at the moment with antisense oligonucleotide therapies for various indications for inherited retinal diseases.
I think I've already got one more question, which is, so you mentioned other retinal diseases that it's being tested on. Are there any inherited macular diseases that antisense oligonucleotide therapy is being tried with?
As far as I'm aware, there have been. I'm not sure, but I think there might be some for age-related macular degeneration, but they are not to treat them. Obviously, that's a complex disease, which doesn't have a single, genetic variation is associated with. It's not monogenic disease, it's a complex disease. And so a lot of people are thinking about targeting pathways which may be common to macular degenerations, including age-related macular degenerations. But by that I mean pathways which are commonly linked to the association and the formation of the drusen. So rather than, like, our approach is to actually target the disease association variant itself, there are some which have aimed at targeting pathways which are shared across these different related conditions. So there is quite a lot of research going on in that field as well.
That's really interesting. Yeah. Colin, did you have any questions?
Well, to be fair, you asked the first question I was going to ask. You beat me too, which was what are the next steps? So I just find all of this very, very incredible. It's just very exciting. Are you excited about the potential of this, Jackie?
Oh yes, I am. I really am. I'm very, very.
Reach.
Yes, I'm very excited. Obviously, there's a few more things we need to do. We need to see if we can further increase the potency of the antisense oligonucleotide. There's a few more tweaks we can do chemically, for example. And I do believe in the potential of this antisense oligonucleotide therapy. It's just a huge challenge to try to take the next step towards getting this towards the clinic. There are many challenges, but that's not going to hold us back, obviously.
No, of course not. Yeah, absolutely.
Try our best to get it to the next stage. As I said, one of the biggest challenges is the perception of this as being a rare disease.
Yeah.
So it's the cost benefit ratio of getting something like this widely available to patients out there. But my opinion is that yes, it is a rare disease. However, it is the only mutation in patients worldwide which causes Doyne honeycomb retinal dystrophy. So some inherited retinal dystrophies are caused by a gene, but they're caused by many different genetic variations within that gene, whereas this is a monogenic disorder with a single variant that that causes this disease. So yes, all patients worldwide will have this variant. So to me, that's quite powerful. And I guess the other thing is that there have been studies, I mean, to give you an indication, the allele frequency of this in genome AD is 1.23 to the power of 10 to the minus 5. So it's really, really rare. But when we look at the most prevalent variants identified in the UK, for example, in molecularly confirmed cases of inherited retinal disease, actually this particular variant, not the gene, the variant, does fall within the top 20 most prevalent variants. So although it is a rare disease, and conceptually that's something that needs to be overcome when you're looking at the commercialization and the drive towards the clinic, I think there's a strong argument to be made for why this could and should be an appropriate target to try to take forward into the clinic.
Yeah, I agree. I do agree. Sitting here with an inherited retinal disease myself, I do agree. So anyway, it's been fabulous. I mean, very exciting. It's really good to hear about some of the funding that we as the Macular Society supported you with, and where it's got us on this well very exciting journey.
Yes.
So thank you for coming back and telling us all about it.
Yes, thank you so much for having me. It's been such a pleasure.
Brilliant. Okay, well, fab. So that's the end of this one. So we'll be back again at the end of next month with another session on actually another piece of Macular Society research, funny enough, which is coming to an end. So we're looking forward to hearing about that one as well. So for the time being, thanks very much, and we'll chat to you next month. Thanks.
Thank you. Bye.